You know, when a major crime is committed, the police rarely catch the thief in the exact act of stealing the jewels.
Right.
The alarms are blaring, the vault is wide open, and the culprit is usually long gone.
Oh, yeah. The active event is completely over by the time the authorities actually arrive.
Right, but the detectives, they don't just throw their hands up and go home. I mean, they start looking for the shattered glass, uh, the muddy footprints, maybe the fingerprints left on the safe. They're actively hunting for the evidence left behind by the intruder.
And applying that exact idea to clinical laboratory medicine is, well, it's really the perfect lens for this material. Because the human body is the crime scene.
Yeah, and the invading pathogen is the intruder.
Exactly. And the immune system, well, the immune system actually plays two roles here. It's the security guard actively trying to stop the break-in, but it's also the forensics team. It leaves very specific, readable clues behind in the blood.
And the trick, obviously, is figuring out how to read those clues. So, welcome to our deep dive into the source material. Today, we are exploring Mary Louise Turgeon's comprehensive textbook, Immunology and Serology in Laboratory Medicine, 5th edition.
It's a massive text, but so foundational.
It really is. And our mission here is to provide you, yeah, whether you're a medical student prepping for clinical rotations, maybe a young laboratory professional, or honestly, just an insanely curious, self-taught learner, we want to give you a shortcut to mastering the bedrock of laboratory diagnostics.
Because it can be overwhelming.
Oh, totally. We are going to unpack the what, the how, and the why of clinical immunology and, try to translate the heavy jargon into concepts you can actually use in the real world.
Right, because before anyone can effectively run a diagnostic test in a modern hospital lab, you really have to understand the biological machinery you're testing.
You aren't just reading numbers on a printout.
No, not at all. You're measuring microscopic warfare.
And humanity has actually been observing the aftermath of that warfare for centuries. The text actually takes us way back to 430 BC.
Oh, the plague of Athens.
Yeah. During the plague of Athens, the historian Thucydides, he noticed something profound. People who survived the plague and recovered didn't get sick from it a second time. Their bodies had literally learned. They had acquired immunity.
Which is incredible to observe back then. And if you fast forward to the 11th century, you see the Chinese practicing a rudimentary, albeit super risky form of immunization.
Wait, is this the smallpox thing?
Yeah, they were having people inhale dried powders made from the crusts of smallpox lesions.
Wow, literally inhaling infected crusts.
I mean, they were intentionally triggering the immune system. But the true scientific foundation really crystallized in the 1880s. Louis Pasteur, who is, widely considered the father of immunology.
Right, Pasteur.
Yeah, he started doing groundbreaking work on attenuated vaccines, basically using weakened versions of a disease to train the body safely.
Okay, let's unpack this. The textbook defines immunology fundamentally as the body's ability to recognize self from known self, and then to dispose of that non-self material.
That's the core of it, self versus non-self.
And to do that, the immune response is basically divided into two main camps, right? Innate immunity and adaptive immunity. So think of your innate immunity, the first and second lines of defense, as the medieval Castellan general guards.
I like that analogy.
Yeah, they don't check IDs, they just attack anyone who doesn't look like they belong in the castle.
And to put a biological face on those general guards, we are talking about physical barriers, sure, like your skin, but also cellular players like granulocytes.
Neutrophils, for example.
Exactly. Prime example would be neutrophils. These cells perform phagocytosis. In the simplest terms, a neutrophil literally surrounds, engulfs, and digests bacterial intruders right on the spot.
Like microscopic Pac-Man.
Basically, yeah. It is a rapid, completely non-specific response.
But if the general guards get overwhelmed, the castle calls in the specialized forces. And that's the adaptive immune system, your third line of defense.
Right.
In our analogy, this is a highly trained sniper team. They don't just shoot blindly. They learn the exact face of a specific intruder, eliminate them, and then remember that face for decades in case they ever come back.
And the cells running that sniper team are called lymphocytes. So you have T cells, which are responsible for cell-mediated immunity. They directly bind to and destroy infected cells.
And then the B cells.
Right, B cells and their mature form, plasma cells. These are responsible for humoral immunity, meaning they produce the specialized weapons we call antibodies.
And the text refers to those antibodies as soluble mediators, which there's a whole alphabet soup of them.
There really is.
The B cells produce 5 distinct classes of immunoglobulins: IgG, IgM, IgA, IgD, and IgE.
Plus, you have other circulating defenders.
Yeah, like the complement system, which is this cascade of proteins that helps clear pathogens, and acute phase proteins like C-reactive protein or CRP, which, spikes in your blood during acute inflammation.
Yeah.
But with all these different immunoglobulins, how do we actually know which one is doing what?
Well, what's fascinating here is the biological timeline. That's the key to everything, especially for diagnostic testing. When you are exposed to a brand new pathogen, your body mounts a primary response.
And the first to arrive is?
The very first antibody to arrive at the scene is IgM. It is a massive molecule that responds immediately, but its levels drop off relatively quickly.
Okay, so it's the fast responder.
Yeah. Later on in the timeline, IgG takes over.
Yeah.
Ig is the heavy lifter.
Mm.
It's a smaller molecule, so penetrates tissues better, and most importantly, it forms the basis of your long-term immunological memory.
So, meaning if we draw your blood and we find IgM, we know the crime just happened. It's an active recent infection. But if we find a high concentration of IgG and no IgM, the intruder was there a long time ago. Your security team just kept their mugshot on the wall.
That timeline allows clinicians to pinpoint exactly where a patient is in their disease progression. And taking that knowledge into a clinical setting brings us to the medical detective's toolkit.
Right. How do we exploit those predictable antibody behaviors in a laboratory to figure out what is making a patient sick?
Well, before the text even lets us touch a test tube, it heavily emphasizes safety and quality assurance, QA and quality control, QC.
Yeah, I noticed that. It feels like a massive stop sign before the fun stuff. Why is there such an intense focus on calibration and safety before we even look at the patient's blood?
Because the most brilliant diagnostic technology in the world is utterly useless if the sample was compromised before it even reached the machine. This is the concept of pre-analytic error. If a chemical reagent is expired, or, I don't know, an incubator is 2 degrees too cold, or a pipette is miscalibrated, the test will yield a false result. And in laboratory medicine, a wrong answer is often much more dangerous than no answer at all.
That makes total sense. So, once the QA is locked down, the text introduces basic serologic techniques. One of the foundational methods is creating serial dilutions to calculate an antibody titer.
Right, titers.
I know a titer tells us the concentration of an antibody, but how are we actually doing the math in the lab?
So you take a sample of the patient's serum and you dilute it step by step, half serum, half saline.
Yep.
Then you take that mixture and have it again and again. You're creating weaker and weaker concentrations. You test each dilution to see if it still reacts with the target antigen.
And the titer is?
The titer is simply the highest dilution that still produces a visible reaction. If you have to dilute a sample thousands of times and it is still reacting, the patient has a massive concentration of that specific antibody.
That makes perfect sense. We also see simpler methods in the book, like point-of-care testing or POCT. These are the rapid manual tests done at the bedside or even at home.
Like pregnancy tests.
Exactly. The classic example is this solid-phase immunosorbent card pregnancy test, looking for the hormone human chorionic gonadotropin, or HCG.
And those card tests are incredibly convenient. But the core of the laboratory relies on much heavier technology. For instance, we use agglutination methods.
The clumping.
Yeah, if you mix a patient's serum with a specific antigen and the patient has antibodies against it, those antibodies will cross-link with the antigens. It creates this visible physical clumping that you can see under a microscope, or sometimes even with a naked eye.
I also saw a heavy emphasis on electrophoresis. The book says it separates charged molecules in an electrical field, but I'm trying to visualize how that actually helps diagnose a disease.
Imagine making proteins run a race through a dense gel track.
Okay, a microscopic race.
Right. You place the patient's serum at the starting line and apply an electrical current. Because different proteins have different electrical charges and different physical sizes, they get pushed through the gel at different speeds.
So the small ones go faster.
Exactly. Smaller, highly charged proteins sprint to the finish line, while larger ones lag behind. This separates the complex serum into distinct bands.
And why is that useful clinically?
Well, if a patient has certain types of blood cancer, like multiple myeloma, you will see a massive abnormal band of a single type of antibody stuck in one spot on that track.
Oh, wow. Okay. We also dive deep into molecular techniques, specifically blotting, like southern blots for DNA, northern blots for RNA, and western blots for proteins.
Right.
The text mentions the western blot has historically been crucial as a confirmatory test for HIV. But how does spreading proteins on a gel prove someone has a virus?
It actually builds on that electrophoresis race we
Oh,
just talked about.
like,
You take the actual proteins that make up the HIV virus and you run them through the gel track, separating them by size.
okay, the viral proteins are on the track.
Then you transfer or blot those separated viral proteins onto a membrane. Finally, you wash the patient's blood serum over that membrane. If the patient has HIV, their antibodies will stick to those specific viral proteins.
So the antibodies hunt them down on the membrane.
Exactly. Then we add a specialized chemical that changes color wherever an antibody got stuck. It creates a visible barcode. If the barcode matches the known protein signature of HIV, the diagnosis is confirmed.
That is wild.
Yeah.
And if we need to look at whole cells instead of just proteins, the book details flow cell cytometry, which honestly sounds like science fiction.
It really does.
You shoot lasers at single cells passing through a microscopic tube in single file. And by analyzing how the laser light scatters off the cell, and by attaching fluorescent tags to specific antibodies, the machine can literally count specific types of cells by the thousands per second.
It's called immunophenotyping.
Yeah, and that's how labs count CD4 T cells to monitor the immune systems of patients with AIDS, right?
The precision is just astonishing.
But it makes me wonder, if we have incredibly advanced machines shooting lasers for flow cytometry and fully automated systems running molecular probes, what is the actual role of the human laboratory technician today? Aren't they just loading samples and pushing a button?
It's a common misconception. But the machine is merely a tool. It completely lacks clinical judgment.
Right.
An automated machine can spit out an electrophoretic spike on a graph or flag a bizarre cell count, but it takes a highly trained human mind to recognize patterns of error. A machine cannot synthesize raw data with a patient's clinical history to realize that, hey, this bizarre result is actually due to a specific medication the patient is taking.
So the data is generated by automation, but the medicine is practiced by humans.
Perfectly said.
Well, that detective work is crucial when we take these tools to the front lines. Let's look at how we deploy these tests against real-world infectious diseases. The text uses syphilis, caused by the bacterium Treponema pallidum, as a prime example.
Syphilis testing is a classic two-step dance.
Right. We start with non-treponemal methods like the RPR or VDRL tests.
Yeah. Yes. And those initial screening tests don't actually look for the syphilis bacteria itself. They look for antibodies against substances released by cells that have been damaged by the infection.
Oh, interesting.
Because they're looking at cellular damage, they are very cheap and easy to run on a large scale. But they can yield biological false positives.
Meaning a patient with an autoimmune disease like lupus, or sometimes even just an older, healthy individual, might trigger a positive result on an RPR screen, even though they absolutely don't have syphilis.
Exactly.
So why do we even use them? Why not just use the highly accurate confirmatory test right from the start?
Pure logistics and cost.
Yeah.
The confirmatory tests, the treponemal methods like the FTA test, are complex and very expensive.
What makes them so complex?
The FTA uses fluorescent antibodies that only bind to the actual syphilis bacteria. You don't want to run that highly specialized test on 10,000 healthy people. You use the cheap screen to cast a wide net. And you only use the expensive specific test on the handful of samples that actually get caught in that net.
Ah, that makes sense. Another fascinating diagnostic evaluation in the text is for infectious mononucleosis, famously caused by the Epstein-Barr virus. Here's where it gets really interesting. The lab relies on something called the Davidson differential test.
Oh, yes.
When I read how this works, it honestly felt like a biological sorting hat.
The underlying mechanism is brilliant. Patients with mono develop something called heterophile antibodies. These are bizarre antibodies that react with antigens from entirely different animal species.
Which is weird to begin with.
Very. The problem is people with other random conditions like serum sickness also make heterophile antibodies.
So how does the test tell them apart?
Through a really strict process of elimination using animal red blood cells, we take the patient's serum and expose it to guinea pig kidney cells.
So guinea pig cell. Oh,
Right.
all right.
Guinea pig cells will absorb the antibodies caused by serum sickness, but they ignore the antibodies caused by mono. Then we do another test using beef erythrocytes, which do the exact opposite.
So by seeing which animal cells absorb the antibodies out of the fluid, we basically process of elimination our way to proving the remaining antibodies are specifically reacting to mono.
Yes. And we finally confirm it by dropping in sheep red blood cells and watching them clump together.
It's such an elegant piece of deductive reasoning. You literally force the antibodies to reveal their true identity based on what they refuse to stick to.
It's lab work at its finest.
Another crucial area of infectious testing is the TORCH panel, which is frequently ordered for pregnant women. TORCH stands for toxoplasmosis, rubella, cytomegalovirus, and herpes.
And if we connect this to the bigger picture, back to our earlier discussion about the biological timeline, the distinction between IgM and IgG is never more critical than in a TORCH panel.
Because IgM is the immediate responder and IgG is the long-term memory.
Precisely. If a pregnant patient's blood shows IgM antibodies for rubella, it indicates an acute active infection.
Which is dangerous.
Very. The virus is currently replicating, which means it could cross the placenta and immediately threaten the developing fetus, potentially causing severe congenital defects. Medical intervention is required right then and there.
But what if the lab only finds IgG?
If the lab only finds IgG and no IgM, the clinical picture completely flips. It simply means the mother had a past exposure to rubella, or maybe she was vaccinated and she already possesses existing immunity.
So that IgG is actively protecting both her and the fetus.
Exactly. The exact same antibody test, just looking at a different molecular weight class, completely changes whether the doctor panics or breathes a huge sigh of relief.
So if we want the protective power of that IgG blueprint without risking the devastation of the actual infection, how do we hack the system? That actually brings us to chapter 16, which is an entire primer on vaccines.
A vital chapter.
A vaccine is basically a safe stand-in for the pathogen. It simulates a primary immune response, tricking the body into deploying that early IgM and eventually building the memory IgG forces.
It provides the sniper team with the mugshot before the intruder ever steps foot on the property. When the actual live infection finally shows up, the host is fully prepared with a rapid, overwhelming secondary response.
We've spent this entire deep dive marveling at how brilliantly the immune system defends us. But we really need to look at what happens when the shield becomes the sword.
Yeah, it's not always perfect.
What happens when this incredibly complex machinery misfires and targets the host's own tissues?
The text categorizes these misfires, starting with hypersensitivity reactions, which exist on a spectrum. Type I is your immediate IgE-mediated reaction.
That's allergies.
Right. This is your classic pollen allergy, all the way up to life-threatening anaphylaxis from peanut. On the other end, you have type 4, which is a delayed cell-mediated reaction.
So not antibodies.
No, this isn't driven by antibodies, it's
Yeah.
driven by T cells. It's the reason you don't get a rash until a day or two after you touch poison ivy.
But the truly systemic, devastating misfires are the autoimmune disorders. Let's look at systemic lupus erythematosus, or SLE. The hallmark laboratory test for lupus is looking for anti-nuclear antibodies, or ANA.
Yes, ANA testing.
To do this, labs use indirect immunofluorescence on Hep2 cells. To translate that jargon, we take a slide covered in specially grown human cells, wash the patient's serum over it, and use a fluorescent tag. We are literally looking under a microscope to see if the patient's antibodies are glowing and actively attacking the nuclei of these test cells.
There's also a rapid slide test for lupus using DNP-coated latex particles. You basically coat tiny plastic beads with this specific antigen. If the patient has the autoantibodies, they attack the beads, causing them to physically clump together into a visible grainy texture right on the slide.
And then there is rheumatoid arthritis, or RA. The lab looks for rheumatoid factor, which is wild because rheumatoid factor is an autoantibody directed against the patient's own human IgG.
It is literally an antibody attacking another antibody.
But here is where I get a bit tripped up. So what does this all mean? The text notes that rheumatoid factor can be positive in healthy older individuals or in people just recovering from viral infections. If a test is that broad and can be triggered just by aging, how is it actually useful to a doctor trying to diagnose arthritis?
This raises an important question about the predictive value of any laboratory test, and it really highlights a core belief in medicine. Clinical lab results must never be read in a vacuum.
Right.
A positive rheumatoid factor test is a clue, not a conviction. If a 70-year-old patient has a positive RF test but has zero joint pain, it is likely just an artifact of their aging immune system.
Like the forensics team found a muddy footprint, but there was no actual break-in.
Exactly that. But if that same positive lab test is combined with symmetrical joint inflammation, severe morning stiffness, and a family history, then that generic clue becomes a highly specific, powerful diagnostic tool.
It proves how interconnected the immune network really is and why human clinicians are absolutely required to interpret the data. And that network complexity peaks when we try to manipulate it intentionally, like in transplantation. When a surgeon performs a solid organ or a bone marrow transplant, they are intentionally introducing massive amounts of non-self tissue into a patient.
Which the immune system hates.
Right. To stop the general guards and the sniper teams from destroying the new kidney or in bone marrow transplants, to stop the new immune cells from attacking the patient's body, which is graft versus host disease, we perform rigorous human leukocyte antigen, or HLA, typing.
We're looking at the specific molecular markers on the surface of the cells to match the donor and recipient as closely as genetically possible.
And even with a good match, we still have to chemically suppress the patient's immune system.
Always. And to ensure we haven't suppressed them too much, we use specialized assays like the immuno-ATP assay. This test measures the actual cellular energy, the ATP produced by the patient's T cells. It tells us exactly how functional their immune response currently is.
We even use immunology to fight cancer, searching the blood for specific tumor markers like prostate-specific antigen or PSA to monitor prostate cancer. I mean, the modern laboratory has found a way to track the clues left by almost every disease process imaginable.
They really have. And looking at where this field is heading provides a really profound thought to leave you with. Historically, we learned to harness this system when Georges Kohler engineered monoclonal antibodies.
Right, the hybridomas.
Yeah, he fused sheep red blood cells with cancerous myeloma cells to create a hybridoma, a functionally immortal cell line pumping out one specific perfect antibody in a lab. Today, we aren't just making antibodies, we are mapping HLA genetics down to the exact molecular nucleotide.
We're moving way past just reading the clues left at the crime scene.
We are entering an era of precision immunology. Think about the autoimmune diseases we discuss, like lupus or rheumatoid arthritis. The current standard of care often relies on blunt immunosuppressive drugs,
Which have huge side effects.
because we essentially turn off the entire security system. It stops the autoimmune attack, sure, but it leaves the patient highly vulnerable to basic infections.
Oh.
But as we learn to engineer specific antibodies and decode immune genetics, we're approaching a day where we don't just blindly suppress the immune system.
Wow.
We might literally reprogram the patient's immune software at the DNA level, fixing the misfire without dropping the shield. It is a thrilling frontier.
From the Chinese inhaling smallpox crusts in the 11th century to reprogramming DNA in the 21st, it is an incredible journey.
Absolutely.
Now, before we sign off, to ensure we've fully accomplished our mission of mastering this material, here is a short clinical exercise to test what you've learned today.
Test your knowledge.
Imagine a 20-year-old patient presents to the clinic with extreme fatigue, a sore throat, and swollen lymph nodes. Based on the clinical picture, you suspect infectious mononucleosis. Based on today's deep dive, what specific class of immunoglobulin IgG or IgM would you expect to find elevated in their blood right now to indicate an active infection? And what classic, elegant serologic test using animal red blood cells could you run to definitively confirm the presence of those bizarre heterophile antibodies? Think it over, trace the mechanisms we discussed, and check your notes.
The answers are always there if you just follow the evidence. Thank you for joining us on this exploration of the microscopic battleground of your own body.
Remember, the crime scene of disease is always leaving clues. You just have to know which forensic tools to pull from your toolkit, and most importantly, keep questioning what the evidence is actually telling you. Keep exploring, keep learning, and we'll catch you next time.